Once we have those wastewater streams collected, what can we do to turn those into fertilizer products? So to open up, we're going to be getting a presentation from Nebiyat Nigusie Woldeyohannis. Nebiyat is a PhD student at Addis Ababa University in Professor Addis' laboratory since 2016. And today, Nebiyat will be discussing antimicrobial resistant genes in an investigation into their persistence when considering struvite precipitation from source-separated urine. And I believe Nebiyat has a video for us to look at. So yeah, take it away.
I am going to present about the fate of antibiotics resistance genes and their carriers in human urine-derived struvite. The contents of my presentation are this. First, I will introduce about struvite, then I will explain the methods, thereafter the results and discussion, at last the conclusion, recommendation, and acknowledgements. It's known that struvite can be made from hydrolyzed urine and magnesium salts are added to enhance recovery of nutrients like nitrogen and phosphorus. Struvite is an eco-friendly fertilizer because it releases its nutrients equivalent to the plant's uptake rate. Though World WHO reported that urine is safe to use as a fertilizer after storage of 6 months, nothing has been reported about the fate of ARGs and mobilums during urine-derived fertilizer production, such as struvite. Therefore, this study is aimed at filling this gap by assessing the fate of ARGs and mobilums during struvite production. To do this, we followed this methodology where source-separated urine was collected from passerbys from downtown Addis Ababa city using frame-shift urinals. The collected urine was then mixed and was stored for about 20 days to sanitize. This was followed by struvite production by adding magnesium chloride to enhance recovery of nutrients. Then DNA was extracted from struvite, fresh urine, and stored urine. Then the DNA was sequenced using shotgun sequencing, after which the data was processed to get metagenome-derived genome sequences. Finally, the sequence was passed through Abricate and RasFinder to trace ARGs. On the other hand, to trace mobilums, a machine learning tool called RF Plasmid was used for plasmid prediction, and a binner called bacterial and viral bioinformatics resource was used for fetch. And we have found about 42 clinically important metagenomic-derived sequences. Among these, we have found ARGs of carbapenem aminoglycoside, chloramphenicol, erythromycin, and efluximab to be carried by the microorganisms in both fresh urine, stored urine, and struvite. Critical priority microorganisms were among them. There were also present such as Aeromonas, Acinetobacter, and Enterococcus species were found to carry ARGs. Among these, we can mention Enterococcus faecalis, Aeromonas hydrophila, and Acinetobacter baumannii. Furthermore, we have also identified that the ARGs were laden on mobilums such as fats and plasmids. Here on the plasmids, different ARGs were found, for example, carbapenems, colistin, erythromycin, vancomycin, and other ARGs were carried. On the other hand, ARGs for one of the last results called daptomycin was found. On the other hand, on fats, carbapenems, aminoglycoside, and efluximab, ARGs were found to persist in struvite, and among these, carbapenems ARGs underwent a shift from one ARG type in fresh urine into 3 types of struvite, keeping ARG family the same. Furthermore, during analysis 2 types of fats, we have found Bacillus, Escherichia, and Streptococcus fats to be dominant in fresh urine. On the contrary, in stored urine, in stored urine and struvite, only Streptococcus fats were abundant, more than 50%. This indirectly entails that the dominance of the carrier microorganisms. From this study, fresh urine, stored urine, and struvite contained clinically important bacteria which carried ARGs, and these ARGs were carried by both fats and plasmids. Then we recommend further standardization of the struvite production process with regard to minimization of resistance gene carriers. At last, I would like to thank you for your attention. If you are interested to read more about the study, you can use the following link. Finally, I would like to thank my institute, Addis Ababa University, and USAID for supporting the study.
Issue of antimicrobial resistance with respect to urine-derived fertilizers. If you have any questions on Zoom, please put those into the chat, making sure you can put at Nebiyat before those questions that are directed to her. And yes, moving on now, we will have John Culpepper speaking. John Culpepper co-founded Compost for Good to help communities around the world upcycle all appropriate organic materials to reduce greenhouse gas emissions, keep resources in local communities, create business opportunities, help solve water quality issues, and lower the cost of food and fiber production. John has worked in education, facilities management, and research. And today, John will be discussing Compost for Good's research into integrating urine into a high-temperature compost process. So, John, hopefully you can share your screen. It looks like you're set to go. And yeah, please take it away.
Thank you. And can you hear me?
Yes, wonderfully.
Fantastic. It is a delight to be with you here today. Good afternoon to those of you in Vermont. Good morning, good evening to the rest of you around the world. A little bit about who we are. We are three individuals, myself, my daughter, and a professional grant writer. We partner with not-for-profits in upstate New York. We're funded almost exclusively through grants. And we work with entrepreneurs, policymakers, land managers, all sorts of folks to help them understand that there is a world of organics out there that can be turned into a high-value soil amendment. And that soil amendment has multiple benefits for working landscapes. We also create designs for composting equipment. These designs are open source. They're free and available to anyone who wants them. And a small part of our work is doing research composting urine through a high-temperature composting regimen. So we all know that there are a lot of ways to stabilize plant nutrients from urine. Why on earth would you compost it? Well, we're interested in more than just the plant nutrients that are contained. We use diverted urine as a nitrogenous material from which to make a high-value, biologically robust compost. And we use that to help farmers and other land managers build long-term soil health or soil organic matter, and therefore long-term soil health. We do this also to break down pharmaceuticals and to reduce or eliminate traditional farming inputs, hopefully saving farmers money. Some of the aspects of our work with urine: we've established a modest human urine research and demonstration facility. In that facility, we do recipe development using 2 different processes at least, creating about a cubic yard per batch. We design equipment to process urine at the neighborhood or community scale. We analyze the finished compost in the lab in our lab in upstate New York for beneficial soil microbes. We work with all sorts of folks, policymakers, engineers, farmers, other land managers. Our successes to date: we've processed over 1,300 gallons of diverted human urine in these processes. We've achieved mature compost that meets or exceeds US EPA's Class A guidelines for biosolids compost. That means no restrictions on use. We've created many cubic yards of this material. We've experimented with a variety of ratios between carbon and nitrogen. We've used our compost to inoculate other compost operations in upstate New York to help them build a more robust soil, beneficial soil biology in their compost that they can then transfer onto their soils. And we work with others in the US and other countries to replicate our successes. Most of us know that the world soils are in decline. The UN estimates about a third of the world's are seriously degraded. Soils used to produce food and fiber have been in decline for a long time. And in my judgment, this is due to the loss of soil organic material, over-tillage, and compounds that negatively impact soil microbes. All the while, the world's population is increasing and farmers are paying more money for their inputs. So a little bit of compost soil health 101. Bacteria and fungi, the lowest trophic level organisms in the soil or in compost, break down organic matter and literally minerals, sand, silt, and clay, and extract from those minerals plant nutrients. Those organisms also create biological glues. And by the way, those two organisms are just the first trophic level. You need the second, third, and additional trophic levels to do the real nutrient cycling that happens in productive soils. But those bacteria and fungi, they bind soil components together, creating more pore space and what farmers like to call as good soil tilth. This reduces nutrient and sediment runoff. Healthy soils are more productive. That's intuitive. Biologically robust soils have implications for mitigating climate change. The world's largest carbon sink are the world's oceans. Second to that are the world's soils. Soils can be either carbon emitters or carbon sinks, depending upon the amount of primarily fungi, dead and living fractions of fungi in soil. So from our opinion, the potential benefits of composting urine create local jobs, keep dollars in the local communities, create a more circular plant nutrient economy, reduce harm to downstream biota from wastewater treatment plants, help land managers reduce inputs and therefore save money. And we do this to, we believe that we can speed up the decomposition of other organics. For example, there's a big push in the United States to compost food scraps, and we've demonstrated that our urine recipe using a urine and a carbon bulking material mixed with a composting recipe for food scraps increases the rate of decomposition. And we believe that this will improve water quality by binding up nitrogen phosphorus in a form that's less likely to end up where they shouldn't be. Increase soil organic matter and the microflora associated with healthy soils. Some of the upcoming work that we have in mind, we currently use our compost in qualitative plant trials, but we're looking to partner with others to do more quantitative analyses. We continue to experiment with various recipes to find the best mixes of carbon and nitrogen for the fastest, best compost. We use our herd facility to educate and inspire others. That's the Human Urine Research and Demonstration Facility. And we work with organizations and others who might benefit. In particular, we're working with organizations that have remote campsites and hoping to get them to compost at least the urine fraction of human waste along with food scraps. So, primary takeaway from this talk is that diverted human urine can be processed easily and safely through a high temperature composting regimen to produce a high value and biologically robust compost, which can build long-term soil health. And just FYI, to put a bit of economics in this, there's a company in California doing a big business selling biologically robust compost, 40-pound bags for $200. That does not include shipping. So there's a financial incentive. I would love to connect with anyone who has questions or concerns about this. And here's my contact information. Thank you very much for your attention. I appreciate it.
Okay. Yeah, thanks for kind of illuminating more about the human food nutrient cycles and integrating urine diversion with composting. Up next, we have Harold Leverenz. Harold Leverenz is a research engineer at UC Davis and wastewater process engineer with Biohabitats. Harold co-founded Advanced Environmental Methods LLC in 2015 to support the implementation of nutrient recovery projects. Harold is a registered engineer in California. And today, Harold will be discussing processing source-separated urine from a highway rest area in California. So yes, I see Harold started screen sharing, and yeah, please take it away.
I'm happy to be here today to talk with you folks about some experiments and research that we did at a highway rest area. This is a highway rest area that's located in California on Highway 5, which is a kind of a north-south highway that goes along the west coast of the US. And so it's quite busy, and we collected urine here for a year-long period. I want to go over the results of that, and also just mention that this is the system that we tested kind of as a predecessor to the PAE system. So there's a lot of similar operational experience here that we want to talk about. Just quickly, I wanted to acknowledge that there's a lot of folks who have contributed to this research, especially for this research in particular at this rest area. I highlighted these folks in bold. Russ Adams is my kind of business partner in this endeavor and kind of dedicated to urine fertilizers. Kahui Lim, Jessica Hazard, Thomas Miller, and Zia Jang are all grad students at UC Davis at one point in the time. And the California Department of Transportation really facilitated this research. The Dunnigan, California pilot was conducted at the Dunnigan rest area. So this is basically a bathroom facility for the public located along the highway. And so you can see the building here shown with a brown roof as the bathroom. It basically has several toilets and urinals for men and toilets also for women. And so in this case, we just collected urine from the men's urinals. And we estimated the usage to be about 500 to 1,000 men per day using the urinals. Circled in orange is the pilot facility. So we basically pumped urine from this building with the brown roof underground through a 1-inch tube, polyethylene tube, to this area that's circled. And that's where the pilot was located at. So that was about a distance of about 300 feet or 100 meters. Just to get a sense of what it looked like, we basically put in like piping to intercept the urine from the urinals. And then we put it into we use a pump basically to pump it over to the test facility. So we did pump urine through this 1-inch tube. And there was a few lessons learned there about potential fouling and whatnot, and we'll talk about that. Just to get a sense of what our urine collection and urine diversion system looked like. Like I said, maybe in 2015, we retrofitted this facility and we installed waterless urinals and low flow toilets. And just that action alone reduced the water usage at this facility by 60%. And in a follow-up study, we came back and we basically intercepted the plumbing and drained the urine from the urinals into like a separate pump tank, like a 5-gallon pump tank. And then we pumped the urine using a little diaphragm pump through this 1-inch tubing all the way to where we had urine storage tanks. And we will get into how we configured these things. Just a couple notes on the urine diversion in particular. And what I wanted to show here is just that we experienced quite a bit of clogging in these waterless urinals. Since then, we've gone and we've done a pretty significant review on all the different types of waterless urinals that are on the market in the US. We found that there's a lot of difference. Some models work really well. Some models we had lots of issues with. Retrofitting plumbing for urine collection, we had a lot of issues with, primarily due to lack of slope and poor plumbing installation. But we did notice a few trends. One is that we noticed the mechanism of clogging in these pipes, especially when you have like a liquid trap, that the liquid trap always results in a lot of fouling in the trap and accumulation of struvite and other biomineral accumulation. But we also found that hair is really like hair from people is really the kind of probably the main cause of like starting these clogs in the piping. And so we really think that removing hair and hair control is really critical. But you can see the type of clogging that we get in the pipes, basically just slow accumulation. And these pipes would clog every few months. I also show in the top right corner an image of like a trap that's like a cartridge trap that we find in waterless urinals. And these, we have a lot of issues with fouling. This is after a couple of weeks in the rest area. And you can see pretty significant fouling starting to occur. And another type of trap that's located as an alternative to the liquid trap in the bottom right corner is basically like a silicone check valve. And these silicone valves we found to be a much better replacement to the liquid trap. And we haven't had very significant fouling issues with these and much better performance. So we're also considering what's the best way to even collect urine in the first place. When we drained urine from the urinals, we basically had to go straight into a 5-gallon tank that's shown on the left there. And then we would basically pump the urine out of this tank and into our collection system for processing downstream. I wanted to mention here that we had really significant fouling on all of this urine collection equipment. You can kind of see it in the tank that's on the left, that basically after a week, we would get maybe like 2 liters of struvite and biomineral precipitates. And basically, it seemed to us that it was critical to have this kind of like precipitation tank to allow a place for all those minerals to fall out. I should mention that this is what happens when you don't prevent the hydrolysis reaction. I know we've seen some examples of using acids to prevent hydrolysis, which is a great concept. But if you don't prevent hydrolysis and you just want to use the hydrolysis or accommodate hydrolysis, you can see that there's going to be a lot of biofouling to deal with. And so we would have to go in and clean the system out about once a week to keep it operational. In the center is our level switch, which could control the pump to turn the pump on and off. We didn't have any issues with it, but just to give an example, the type of like fouling and crusting that we had on this device. And then on the right is just an example of measuring the struvite precipitation. And in essence, what we found is that all the calcium and magnesium precipitates out at this point. And basically, once the calcium magnesium is precipitated out, then we don't really have too many fouling issues after that. From there, we pump that liquid, like I said, through the 100 meters of underground polytubing. We did, after a year, we did do an acid cleaning on that tube, and there was a lot of fouling in there, to be expected, I guess. We then took the urine and we pumped it into these 3 tanks in series. This is primarily for hydrolysis and urine storage. So it'd go into the first tank, it would come into the bottom of the first tank and flow up to the top. These are 1 cubic meter tanks, about 200, close to 300 gallon, and then flow from the first tank to the right to the next tank and flow up through that tank. So these were basically upflow storage tanks. And I did place a packing inside just to help encourage, hydrolysis and any particle separation. But I think the real critical item here I wanted to mention is we always keep an oil layer like on these tanks using vegetable oil, maybe like a centimeter thick or so. And the reason there is to prevent ammonia evaporation. And basically what we find is that in all these systems, we can get really significant ammonia evaporation if you don't try to prevent it. And so here we basically use oil caps on all of the, open, wherever there's an open surface to the atmosphere, we use oil to help retain the ammonia so it doesn't evaporate. In terms of how much urine we collected over the year, we basically found that we were getting about 30 gallons a day on average. And you can see after a year, we had collected about 11,000 gallon, which I think, about 11,000 gallons. So close to 1,000 gallons a month we were collecting. So maybe was that about 4 cubic meter per 40 cubic meter? In terms of the process, this is similar to the process we looked at for the PAE system. And really what we looked at this as a coupled system where we have both steam distillation and struvite precipitation. We put the steam distillation first because it's a thermal process that operates around 95 C. So we get really good pasteurization. And so basically after this pasteurization step, we've also extracted the ammonia. And then we remove the phosphate using a precipitation reaction by adding magnesium. And I think the one thing I wanted to mention here is that in the steam distillation process, there's like a few like dials you can turn to make this system work better. For example, if you turn up the heat injection, it basically removes more ammonia. And that was one of the variables we were looking at here, is that how does this system work if we extract the ammonia upstream? What impact does that have on the struvite production downstream? And that's the question we want to look at today. I just want to mention that this system has a capacity of about 2 cubic meter per day, or about 500 gallons, 500 to 600 gallons per day in total. It could be scaled up, but for this, which is a 6-inch column or 150 millimeter, then we basically can operate at 500 gallon per day, roughly. This is a view of the pilot system. We basically built this system with glass so that we could keep an eye on how it works internally. And so you can see here in the center is the column. So this is an 8-foot glass column with a stainless steel high surface area packing. And that's basically to separate ammonia from urine. And then you can see on the right, we have a couple tanks for storing the distilled ammonium and the struvite that we collect. The struvite we just make straight in the tank. So just looking at the kind of the efficiency of ammonia collection, that overall we were basically collecting about, maybe 20 to 30 kilograms of nitrogen as ammonium each month, with a total of maybe we collected about 230 kilograms total after a year. And I should mention that because this was an experiment, we were testing a bunch of different operational configurations, that this doesn't represent like that maybe we can only, recover 80% of the ammonium. It's just that this was kind of more of an experiment. They were looking at different operations. And in this case, we recovered 227 kilograms out of about 290 kilograms that was potential. All in all, about 1,600 liters of distillate, or gallons, sorry. The struvite we collected, like I said, as a struvite powder. And basically, in terms of total phosphate recovered, we did recover quite a bit of it. But again, like I said, this was an experiment. And so we were really testing different configurations, and this may not represent the best operational performance. After the struvite was dried, you can see it just comes out like a brick out of the oven. And that can be crumbled pretty easily. Some of the variables that we were looking at in the study is the most key variable is the feed to steam ratio that really affects how much energy gets put into this process and affects overall performance. The process pH, we generally keep it around pH 9. And I wanted to talk about these different alkalinity sources that we use, both sodium-based alkalinity like sodium carbonate and potassium-based alkalinity sources. What we found is that when we're basically making struvite, if we use a type of a sodium-based alkalinity source upstream, like sodium carbonate, that increases the amount of sodium in the struvite. And if we use a type of a potassium alkalinity source upstream, it increased the amount of potassium in the struvite. And so what we found is that the struvite we were making was always a blend of a sodium struvite, a potassium struvite, and an ammonium struvite. And that basically, depending on how we operated the upstream process, we could really control the composition of the struvite that was forming. And so this is the summary from basically this whole study, is that if we operate at a low feed to steam ratio, which basically means higher energy input, if we put more steam into the process, we could strip out all of the ammonia, like 100% of the ammonium. But if we strip out all the ammonium, then there's no ammonium left to make struvite, to make ammonium struvite. And therefore we get basically poorer phosphorus recovery, high energy input, very high ammonium recovery. And because the ammonium's been stripped out, it makes basically preferentially potassium struvite. If we operate at a medium feed to steam ratio, which is kind of like a moderate energy input, we leave some ammonium in the liquid. We basically make a really high fraction of ammonium struvite. And similarly, if we operate at a high feed to steam ratio, which means relatively low energy input, kind of a lazy strip, then we leave a lot of ammonia in there and we maximize struvite production, but we don't recover too much potassium. And I should mention that we never recover as much potassium as we added. And so trying to make potassium struvite in this configuration doesn't really make a lot of sense. And with that, I wanted to mention that this paper is available, published a few months ago. And so you could collect it at this link, or you can search for it. It's open access, so it should be easy to get. And if you need to contact me, my email addresses are here. AEM is the supplier of nutrient recovery equipment, and Biohabitats is a collaborator in installing these systems. So with that, I'd like to thank you for your attention and happy to answer any questions when we get to the question and answer period. Thanks.
Thank you so much, Harold, for that discussion of source separation at rest areas, which is as many have noted, quite a potent opportunity for us. Coming up next is Dr. Veera Koskue. Dr. Veera Koskue works as a postdoctoral research fellow at the Department of Chemical Engineering, University of Melbourne, Australia. She has several years of research experience on nutrient recovery from different wastewater fractions using bioelectrochemical and electrochemical methods. She is currently involved in the interdisciplinary Nutrients in a Circular Economy Research Hub funded by the Australian Research Council and industry partners. And today, Veera will be discussing the UGold bioelectroconcentration process for urine-derived fertilizer production. Please go ahead, Dr. Koskue.
Thank you. I hope you can hear me all right.
Yes.
Fantastic. So yes, good morning from Australia. Excuse the terrible lighting. It's around 4 a.m. here. But as mentioned, my name is Veera Koskue. I work as a postdoctoral research fellow at the University of Melbourne, but I'm actually currently in Brisbane, where I'm about to start our pilot scale experiments using our UGold technology that I will be introducing you to in this presentation. And to give you a bit of context, as mentioned, our project is being carried out as part of the Nutrients in a Circular Economy, or NICE, research hub that's partly funded by the Australian Research Council. So I'm working closely together with my supervisor, Stefano from Uni Melbourne, and also a number of project partners. And just before I get into the actual topic, I would also like to take a moment to acknowledge the First Nations peoples of Australia as the traditional owners of the land on which I have the privilege to live and work today. So more specifically, that's the Turrbal and Yagera people here in Brisbane, and also the Wurundjeri people of the Kulin Nation in what is now known as Melbourne, where most of this work has been carried out. So as I mentioned, the technology we've developed is, or we call it UGold, and it's based on bioelectroconcentration. And that essentially means operating with a 3-chamber electrochemical system where the anodic oxidation reactions are facilitated by electroactive microorganisms. So these bacteria are able to use some of the chemical energy in hydrolyzed urine, mostly present as acetate, and convert that into electrical energy. And this electricity or we do supply some external electricity as well, and that is used as a driving force for cations and anions, including ammonium nitrogen and potassium as cations, and phosphorus as phosphate, an anion, to migrate into this recovery chamber placed in between the anode and cathode and separated from them by ion exchange membranes. So in the end, we produce a concentrated liquid fertilizer containing all these key nutrients we're interested in recovering. At the cathode, at the same time, the protons and electrons are reduced to hydrogen gas. And if that is collected separately, that can also be used as an energy carrier. And as I mentioned, we're currently working on scaling this technology up by a factor of around 20. So from my lab scale experiments in Melbourne. We're now commencing our pilot facility here in Brisbane. If you missed my virtual tour of the pilot yesterday in the conference, I hope you can still view that recording later. But as I mentioned, we're increasing the factor of both the reactor volume and the treatment capacity by approximately 20 times. And in preparation for the pilot, there were still a few technical and operational bottlenecks that I was addressing in my lab experiments in these past few months. And that is what I will be talking to you today about, more specifically focusing on two specific aspects. Firstly, enriching this kind of an electroactive biomass at the anode can, or in the past has proven to be challenging when using hydrolyzed urine, because the conditions can be a bit harsh, mostly because of the total ammonium nitrogen or TAN concentration, which is typically in the range of several grams per liter. And that, combined with the high pH of the hydrolyzed urine, means that a large fraction of this is present as ammonia, which is especially toxic to a lot of microorganisms. So in the past, what people have been doing is either using a microbial community from a system already operating with hydrolyzed urine as the inoculum, so that it's already used to using urine. But of course, that is not always available. Alternatively, sometimes people can start with an artificial medium with a much lower original TAN concentration, and then gradually increase the concentration, giving the microbial community time to get used to it. This, on the other hand, can take up to several months to complete, which is also not very feasible. The second challenge I was addressing in my experiments was the gas buildup and accumulation in the system. So as you could, as we saw a couple of slides ago, both the oxidation at the anode and the reduction at the cathode generate gases, CO2 at the anode and hydrogen at the cathode. And in the past, we've observed these gases being accumulated in the electrode chambers, replacing the liquid volume, which leads to all kinds of problems. So the other thing I was looking at in my lab experiments was how to mitigate this and make sure the gas exits the system. So as a summary, I firstly looked at finding an effective startup strategy for the bioanode, where we could minimize the time needed to enrich this microbial community. And then secondly, looked at technical solutions to remove the gas from the electrode chambers. So regarding the startup of the bioanode, what we hypothesized was that if we mix the hydrolyzed urine with waste-activated sludge, we could both bring the pH of this initial startup solution down and also dilute the TAN concentration in the very beginning. At the same time, the sludge would also function as a source of a wide range of microorganisms to use as an inoculum for the bioanode. And so what we did was tested a few different ratios of mixing urine and sludge volumetrically, and used the current generation in the system as the performance indicator. And what we saw was that the current generation started within 1 day when inoculated with these mixtures in batch mode. And then when we saw kind of stable current generation, we switched on the pump and started feeding undiluted hydrolyzed urine very slowly in the beginning. But as the response in all these reactors was very positive, and then increased the feed rate and the current generation increased proportionally. And essentially what we saw was that the higher the initial sludge proportion in the startup mixture, the higher the current generation was in these early stages. Regardless, we were able to start the continuous feeding of hydrolyzed urine within 10 days or so, which was very positive news. And then the second bottleneck was the gas accumulation and mitigating that. So what we did was implement these external circulation bottles. So we would pump the analyte and catalyte through these bottles to provide mixing to the chambers, and also allow any gas in the liquid to exit from the top of the bottle or column. And that also proved to be a very efficient way of removing the gas from the system. So we had no issues with gas buildup. And in the pilot, the columns on the right are going to be used for this exact same purpose. So to summarize, I presented these two challenges we had observed in our previous lab experiments and tried to address them in my experiments quite successfully. So firstly, the mixing of hydrolyzed urine with waste activated sludge proved to be a very efficient startup strategy for enriching a electroactive bioanode that was tolerant of continuous feeding of hydrolyzed urine in less than 2 weeks. And secondly, using these external recirculation columns was an effective way of eliminating any gas that was formed in the system. So I'd like to leave you with that, and thank you for your attention. I warmly encourage you to follow our progress on our project website. You can also find the project and myself on LinkedIn, so I'd be happy to connect with anyone. And you can also find my email address there if you have any further inquiries. Thank you very much.
Thank you so much, Veera.
So I will be discussing long-term electrochemical stripping for nitrogen recovery.
Speak up a little for the folks in the back.
Okay, yeah, sure thing. Try to project. Yes, so we're all here working on circularizing our nitrogen economy for strong economic and environmental benefits. Most notably, my lab's interested in reducing the energy consumption, chemical use, and costs associated with both chemical production of fertilizer or other chemical products and treatment of wastewater. Urine and source separated urine is an important foothold in circularizing our nitrogen economies because of its small percent of wastewater by volume and high percent of nitrogen in wastewater. Urine is also highly conductive, which is what makes electrochemical methods of nitrogen removal particularly advantageous. And I'm going to talk about that some more. So, oh, wait, you can see all the, okay. I didn't know you guys couldn't see. How do I close the panels here? Hide video panel and hide.
What are you?
Okay, but for the people in person. Okay. And then close that out. Okay, great. Now everyone can see the screen. So this is a schematic of our electrochemical stripping reactors that we use in our lab. It's a 3-chamber system that's slightly different than what Veera just discussed. So I'm going to walk you through how our electrochemical stripping reactors, or ECS reactors, work. So in the first chamber, we have urine flow in, and this is the anode of our electrochemical cell. You can see with that little bar above. So the anode has a low pH environment, which makes all of the ammonia in the urine convert to its cationic form of ammonium. And as a cation, it can transfer through this cationic exchange membrane that separates chambers 1 and 2. In chamber 2, there is a high pH environment because it's the cathode of the electrochemical cell. And this converts all of the ammonium in its cationic form to neutral ammonia again, which at ambient conditions volatilizes and can pass through the second barrier into the third chamber, because the second barrier is a gas permeable membrane. So in this third chamber, we call this the trap of the system because it's where our ammonia eventually winds up as ammonium sulfate, which is a type of fertilizer. So this is the overall mechanism of our electrochemical stripping, ECS reactors. And I'm going to talk about removal and recovery a lot. So when I say removal in this presentation, I mean everything we get out of the urine flowing through that first chamber. And when I talk about recovery, I mean everything we got all the way into that final chamber, which is our concentrated high purity nitrogen product. So this is a graph made by one of my colleagues about nutrient removal and recovery barriers across technologies. And so the most reported knowledge gaps are in operating conditions, process mechanism, and reactor configurations, all those optimizations that go into those. And while we in our lab are continuing to look at these knowledge gaps, the information I'm going to present to you guys focuses more on these kind of down the line information gaps that we have about the influent effects to ECS performance, long-term performance, and continuous mode. These kind of intermediate steps are also important for even farther down the line knowledge gaps in the techno-economic analysis, the life cycle assessment, and scale-up considerations. So I'm going to talk about two research questions in this presentation. The first is, what urine collection and storage practices maximize nitrogen recovery in ECS systems? And also, can ECS work for long-term and continuous treatment? Previous to our studies, the longest ECS didn't go anywhere near the length of a month, more on the scale of like 24 hours or even 24 hours. So I'm going to talk about these three characteristics that we studied for our impacts to collection and storage practices. The first one is pretty self-explanatory, of flush volume. The next is the divalent ion concentration, which has been talked about in multiple studies previously in this panel. And so this is referring to how magnesium and calcium are ions. They're present in fresh urine as we excrete it, but over time they precipitate into struvite and these other solids that have produced fouling, and we've talked about in this conference so far. And when they precipitate, they are no longer in solution. One of the reasons why we're interested in the divalent ion concentration in our ECS systems is that as cations, they compete with ammonia for transfer across that first cation exchange membrane, which makes us interested in what their presence does for the performance of the system. And then at the end there, we see extent of hydrolysis. A lot of other speakers have talked about that, but just to remind people who don't know, as we excrete urine, there is nitrogen present in the form of urea initially, but over time, that urea is converted through hydrolysis into ammonia. And so that impacts the kind of chemical makeup of our influent urine. And so we're interested in what that extent of hydrolysis does to performance. So we took completely hydrolyzed urine, and we also took different levels of mixed, completely hydrolyzed and mixed with fresh urine, which kind of replicates storage with if it's being mixed with fresh urine or if it's been, stored by itself for a long time. This is a picture just to give like a kind of mental image for people of our ECS reactors. I'm not going to go through everything that's going on here, but if you guys have questions, I would love to talk about this system with you guys afterwards. I'll just highlight these are two of our little ECS reactors. They're not connected. We're just running two at once. And then down there are our influent tanks that go into the systems and then flow right back out after being treated into that middle tank. And so this is what we mean by continuous mode. Instead of batches recirculating in and out and in and out, it's like you go in and then the stuff that comes out does not mix with the stuff that's going in. So it's just continuously going in and out of our system. So we looked at the nitrogen transfer kind of percents and efficiencies, and we also looked at energy consumption for both removal and recovery for 3 different characteristics. So that's 12 graphs that I'm not going to go through the trends of each of these with you guys. That would be way too long and not as interesting. So I will be instead highlighting kind of one key conclusion from our nitrogen transfer results and another from our energy consumption results. The first that I want to drive home about the influent conditions is that even as we changed all these different conditions related to how the urine is collected and stored, we still saw 69 to 96% nitrogen removal and 65 to 88% nitrogen recovery across all these like wacky changes we were making to how we collected the urine and stored it. Now to the energy consumption side of things, I want to point out this graph that shows the energy consumption for a standard flush urine sample and unmodified, which is source separated, or the ultra low flush. And so as we see, the standard flush volume required a much more energy intensive load to treat compared to the other flush volume and all other conditions that we investigated. Zooming in on the y-axis about 5 times so we can parse out the trends of those other conditions, we see that both increasing divalent ion concentration slightly increases energy consumption, and then reducing the extent of hydrolysis more significantly impacts energy consumption. So leaving graph mode and returning to pleasant graphics mode, in flush volume, that means we want to minimize the flush volume for energy efficient urine treatment. We want to ensure that we have fully precipitated the divalent ions in our urine. And we also want to use completely hydrolyzed urine for electrochemical stripping treatment of urine. Now to that second research question of how does ECS work over long-term and in continuous treatment, we used the same setup, but now we ran it for 35 and 37 days in duplicate. And these are the results from that experiment. So on the y-axis here, you'll see the removal percent for that recovery or removal and recovery parameter. And there we see an 83% removal and 73%, I'll change this, recovery. And you can see that it's a pretty stable number too over time when there's a kind of initial period of I don't want to say volatility because it's not actually volatile, but an initial startup period. And then if you look at the right-hand y-axis there for the total ammonia nitrogen concentration, we got 8 batches of about 21 grams per liter ammonium sulfate product. And the numbers above the peaks right there are just the pH it was collected at. So it was at about 8.6, which is relatively neutral, which is also important to know about your fertilizer product. So additionally, beyond just wow, look at our results, we also had other more qualitative findings about the failure modes in long-term ECS and also process monitoring parameters. So in this graph here, the blue dots are the effluent pH of the first chamber, meaning it should be acidic. Like we talked about, that first chamber, we have to have it acidic for the system to work. And so when the pH spiked up, we replaced the CEM and it went back down. So basically this both points to how the CEM failure is how CEM replacement fixed the problem. So it was the CEM that was the problem. And that's the cation exchange membrane to remind people. And also that pH is an inexpensive and pretty effective way of monitoring what's going on in our system. So yeah, returning now to our initial research questions, just to hit this home again. Maybe move this over. We want to minimize flush volume and pre-age our urine for our most energy efficient recovery of nitrogen in ECS. And across all influent conditions, we saw 69 to 96% nitrogen removal, 65 to 88% nitrogen recovery. And for our second research question, we were able to continuously treat urine for over 35 days in a relatively stable and repeatable way with 83% nitrogen removal and 73% nitrogen recovery, which is kind of on that upper end of the range from the varying influent conditions. If you'd like to learn more about all the details that went into these studies, we have a preprint up. So this means that we've written a paper and we're just waiting for it to be published. But while we wait, you can still access the information. That QR code is there if anyone wants to scan it. I'll bring it back up on the thank you slide too. So if you don't get it now, no worries. And to touch on briefly what we're going to talk about next, or next studies in our group with this information. So we're next looking at flexible product recovery from ECS. So basically adding a 4th chamber to our 3-chamber cell to recover an additional product of ammonium hydroxide, which is another just chemical that can add to the kind of value portfolio of what we can get out of treating urine with ECS. Also looking at these failure mechanisms in membrane fouling and how those, what's going on in that essentially. And also that life cycle assessment stuff that is, you need to know how your system's working to be able to calculate all that stuff. And then also integration with other technologies like solar or phosphorus removal. I'd like to thank all of my lab mates and all of the great organizations that are helping us do this research. And yeah, thank you for listening. If you have any questions, I would love to talk more about this.
All right. Wonderful. Great. And I think so we have some time for a few questions right now. I think so if you have any questions, please come up. But the first one we have was from Christina for Dr. Koskue. And that was about, were there any additional permitting requirements that you had to meet in order to incorporate waste activated sludge into your process?
At this stage, no. For research purposes, that's fine. I guess we're not at the stage yet where our product would actually be put on the market. And question, and kind of something we're also trying to, as I mentioned, we're working in this large research hub where we're trying to encompass a lot of the different aspects of recycled fertilizer production. So definitely also looking at the regulatory requirements and aspects of it. But so far, no need.
Cool. Thank you so much. We have Malcolm, yes, please.
This is a question for Veera or really anybody, but I don't quite understand. Do we have an opportunity to recover or to generate energy from urine? Is that what we're doing, or?
Yes, thank you for your question. That is correct. So as I mentioned, there is a lot of chemical energy contained in urine in the form of organics. And so by using bacteria that are able to take that organic matter and then generate electrons that they're able to give out extracellularly. So to a solid electrode, we actually can generate electrical energy from urine. It's not, at least in our system, it's not quite enough to be self-sustained. So there's not enough energy produced that it would be functioning without or completely without any external energy input, but it definitely brings down the energy consumption.
And how much energy do you create?
Oh, that's a tricky one. I don't have any. I can't give you any numbers from the top of my head, unfortunately. But I can look into that. Thanks for your questions.
And I think we have time for maybe one or two more questions, but this one was for Meili, and it was, what are kind of the potential for reducing energy use in the electrochemical system, and what are forgetting the second part, and how long do you think it could operate for, maybe beyond the 35-day?
Yeah. So I think how long it can last beyond the 35-day question is going back to that process optimization stuff of finding the right materials that will last the longest. Right now, the costs for those materials and like the membranes and the upkeep is the greatest cost. It's not really the electricity. And so more working with reactor configuration and material optimization is probably the next step for having longer term projects. Or like whoever asked it, it wasn't actually your question. So yeah, I hope that answers the question.
And one follow-up that someone else asked is what about phosphorus in your process?
Oh, yeah.
For phosphorus.
Totally. So our lab does a lot of kind of elemental resource recovery from wastewater, not just in urine or nitrogen. So we have other projects looking at selective adsorbents, where essentially you run the wastewater through like media, and it selectively removes elements that you're interested in. And so we have some projects looking at phosphorus recovery using that technology. And we have like a grand plan to integrate the different technologies, and it's not that grand. I'm like working on it now. So like we actually have this project that's working on it. And so I guess stay tuned is one answer to that.
Well, thank you so much. Are there any other questions in the crowd or on Zoom? Looks like some are getting answered in the chat as we speak. Otherwise, I guess I'll pass it back over to Julia. Thank you to all the researchers who presented their work.
Yes. Great. Thanks so much, everyone.